Boar taint is that really unpleasant smell and associated taste from eating boar meat. If you eat meat from intact males after they've reached puberty, and we're talking about pigs, it doesn't taste good. And we've known about it. The Romans knew about it, so we've known about it forever. It didn't taste good then, and it still doesn't.
Well, welcome to Swine Ethics Canada. Today I have with me Dr. Trish Berger, who is a distinguished professor emeriti at UC Davis. Welcome, Dr. Berger.
Thank you. It's good to be with you.
So my name is Ruud Zijlstra. I work at University of Alberta in Canada, and today's topic that I have with Trish deals with boar taint. So just as a question to start this topic, what is boar taint and what causes it?
So boar taint is that really unpleasant smell and associated taste from eating boar meat. If you eat meat from intact males after they've reached puberty, and we're talking about pigs, it doesn't taste good. And we've known about it. The Romans knew about it, so we've known about it forever. It didn't taste good then, and it still doesn't.
So just to follow up, if I compare myself to my wife at home, I know who between the two of us would be more subjected to it, because my smell is not as good as my wife's. How bad is the smell and how bad is the taste? Is it something most people really want to avoid, or are some people not even smelling it?
So it varies a lot with individual. Over the population, women are more likely to smell it than men. There are people who don't have the ability to smell it at all. And then there are people for whom they want to leave the room, they want to leave the house. So there is a lot of variability in how unpleasant one perceives it to be.
Wisenetics turns podcast airtime into brand authority. We don't sell ads, we elevate voices. Curious how far your voice can go to become a reference in the industry and attract more leads? Scan the QR code and discover how we can turn your expertise into unmatched brand authority. Let's transform expertise into influence, starting now.
So when you then look at boar taint, what are some of the underlying mechanisms that cause boar taint to occur in these intact boars, as you described?
There's bacteria live in every large intestine. And in the case of pigs, these bacteria are synthesizing scatol. It's the compound that smells from the lagoons, basically, or sewage treatment plants. And they're synthesizing this from tryptophan. It's made in the large intestine, it gets absorbed into the blood, it goes to the liver. And now the question is it metabolized fast enough, or is some of it go to the fat, get deposited in the fat? So when you heat up the fat when you're cooking, you get this horrible smell. In the case of females and barrows, they synthesize scatol because they also have large intestines, they also have microbes. But when the scatol gets to the liver, because it's absorbed into the blood and goes to the liver has these cytochrome P450 enzymes that will metabolize the scatol to metabolites, which are eventually changed into water-soluble compounds and eliminated. But in the case of the boar testes is this amazing steroidogenic factory, and the whole boar reproductive tract can take some of the steroids that are synthesized in the testis and in some cases convert them. So the boar synthesizes androstenone. We've thought that was made in the testis. That's sort of the classical idea, but it may actually have a significant amount made in other parts of the boar reproductive tract. The androstenone in the liver inhibits these CYP cytochrome P450 enzymes, or it's thought to inhibit these cytochrome P450 enzymes. That's been demonstrated in vitro. It has not previously been possible to demonstrate it in vivo. By inhibiting these cytochrome P450 enzymes, we aren't metabolizing scatol in the liver in the boars because we have the androstenone, and then we have scatol accumulating in the fat and smelling.
Interesting. So relative to that, that must obviously be related to the practice we have in the pork industry of castrating boars. So what are some of the advantages and disadvantages of harvesting meat from boars rather than barrows?
So if one harvests meat from boars rather than barrows, one gets that feed advantage, the increased feed efficiency that boars have. Pettigrew, way back in Illinois, I shouldn't say way back in Illinois, but Pettigrew did a sort of a survey of all the papers that compared the efficiency in boars versus barrows. Not every study shows the same rate, but overall it was about a 10% increase in feed efficiency in boars compared to barrows. That's huge, but it doesn't do any good if the boar stinks.
Packing plants, I presume, have a role to play here because they want to perhaps entirely avoid the risk of receiving boars at their plant. So if there is for producers such an advantage, pig producers such an advantage, why are we then still dealing with barrows as opposed to boars? Is it strictly to deal with boar taint?
I would say that we are dealing with barrows rather than boars strictly as a result of boar taint. There's some other ways to get rid of boar taint, but basically, rather than physically castrating the animal, you can immunologically castrate the animal. But if you've immunologically castrated the animal, you've also eliminated that feed efficiency growth advantage of boars.
To some extent, yeah. I agree with that. So it's simply at the slaughter plant, the workers there, they don't want to receive intact boars simply for the risk of having boar taint in pork products. That's basically the only.
One needs to do something because they used to slaughter boars. They tried to slaughter boars at a younger age, younger weight, to avoid boar taint, so you weren't dealing with animals that were in that peripuberal, postpuberal range. But then you're processing smaller animals with the same processing cost per carcass, and that's not an economic.
Yeah, it's not cost effective because you have way less pork per kill, if you like to describe it like that. So then, obviously, you're an expert in this area. How does the research that you did contribute to eliminating the disadvantage of castrating boars?
I'm pretty excited about the research, and I should mention that it's a work in progress. We have some very promising preliminary data, but we don't have everything done to etch things in concrete. So I talked about how androstenone was important to in creating boar taint because it inhibits the cytochrome P450 enzymes in the liver. So we thought if we can eliminate androstenone, then we can basically make these intact boars like sows and barrows. So that's basically what our hypothesis is that we can eliminate the androstenone. The males will still be fertile, they will still grow, they will still be totally healthy, but they will have reduced boar taint, or they won't have boar taint.
Yeah. So how do you go about that? How do you make that happen?
So for us, we used CRISPR-Cas9 to edit the promoter region of the androstenone gene. So the promoter region, it's sort of where all the machinery gathers in order to synthesize the protein. And if we delete some of the DNA in that region, we can't gather the machinery, so we cannot start the process to make the protein that's going to make androstenone.
So how successful have these efforts been? Can you completely then eliminate androstenone or part of it, or what have you observed?
When we do this, we've taken out a 7 base pair portion of the DNA and a 4 base pair portion of the DNA, as well as a 2 base pair and a +1, depending on which chromosome we're talking about in which of our founder animals. We also took out a 16 base pair segment in a very few of the chromosomes in one animal. So in the offspring that we created from our initial animals, we had some males that had a 4 base pair deletion on one chromosome and a 7 base pair deletion on the other chromosome. We also had some that only had a -2, a 2 base pair deletion on one chromosome and a 1 base pair insertion on the other. So we have a range. If we look at the animals overall, they have smaller bulbourethral glands compared to control animals, animals that we haven't done anything to, that are as similar to our experimental animals from the point of view of genetics. We harvested all these animals at 6 months of age to be sure we gave them time for Boar Taint to develop. We've been able to show that the enzyme in one of our accessory sex glands, one of these other tissues in the boar reproductive tract, has, right now I don't remember, I think it's an 8-fold lower activity than the control boars. We looked at this enzyme in the testes, but so many boars, by the time they're this age, don't really have much of this enzyme in the testes anyway. So that's, what it is. It's hard. When you don't have much, it's hard to show that you have even less. We've also, when we've harvested these boars, and we've had somebody who's particularly sensitive to the smell of the carcasses. So all of the control boars had some smell, although one of them was pretty faint. The carcasses from the animals that have the 4 base pair and 7 base pair deletion, she couldn't smell. The others, most of them were faint. There were a couple that were more in that, definitely Boar Taint territory. We are planning a consumer sensory panel to do the taste, but we needed all the students back to have our sensory panel. And we haven't been able to, that hasn't happened yet. It will happen soon, but not yet.
So then based on your description, it is then critical to remove both that 4 and 7 base pair units in order to get the lowest level, or basically to end up with no Boar Taint that you can detect via nose. Would that be a correct description?
One chromosome has 4 base pairs removed, and the other chromosome has 7 base pairs removed. So I think that if we have 7 base pairs removed on both chromosomes, that will be quite effective. We're also in the process of generating some animals that will have 16 base pairs removed on each chromosome. So an even bigger deletion. But it seemed like if you did less than that total of 11 base pairs removed, you didn't get the effect. You probably have to have an effect on both chromosomes, but we don't absolutely know that. Scientists are always a little leery to
Yeah. So in other words, you want to reduce the base pair removal to the minimum, but still be effective to control boar taint.
I don't know if we need to reduce the voice pair or deletion to a minimum. I don't know that, but you work with what experimental models you have.
Sure.
But I do think we need to eliminate it on both chromosomes.
Yeah.
I'd like to mention that these animals look totally healthy. They act totally healthy. We did some chem panel analysis of our initial animals, totally within normal, blood enzyme ranges, and they've been fertile.
So then obviously you have done a lot of work before this experiment to properly identify which chromosome, which base pairs have to be removed. Could you explain a little bit about those efforts? Like, how did you identify what to cut?
So there's some serendipity involved here. The chromosomes in the pig have been mapped. They're not as well mapped as they are in cattle, and definitely not as well mapped as in the mouse or the human. So we had some indication of what the sequence was in this region around the enzyme that we were targeting. When we designed our target, we were actually not going to be targeting the promoter, this region where all the machinery gathers. We were actually going to be targeting this initial part of the enzyme. And by eliminating a part of the initial enzyme, then you don't get the rest of the enzyme, and all's good. Well, it turns out that about the time we were transferring our edited 4 to 8 cell embryos into our recipient, a revision of the pig genome came out, which said that the region that we had targeted was not in the initial part of the actual protein, it was in the promoter region. I wasn't terribly thrilled when I first found that out, but then, okay, if we've eliminated promoter, then if we can eliminate the machinery there,
maybe that works. Yeah.
It works. And, it could be even better. We don't know very much about exactly which regions of the promoter are essential. And maybe every part of the promoter is essential, and maybe part of it is, maybe it can use it. Maybe we don't know that. So we may have had some serendipity in terms of hitting a part of the promoter that actually works.
Well, you can call it good luck. It's also, you still targeted a particular area.
Oh, and, yeah. Well, pat myself on the back. Yeah, we
exactly,
did good.
Exactly, yeah. So when you then look at the topic of boar taint overall and you zoom out a little bit, how big of an issue is it still in the North American pig industry?
Well, if I pick the U.S. as opposed to Canada,
please.
We harvested over 60 million barrows last year. That's a lot of pigs.
Yep.
That is a real loss in feed efficiency.
Yep.
So from the point of view of we think our approach will allow us to get the feed efficiency, there's also the perceived animal welfare concern for castration.
Yep, exactly. Yep.
There are some alternatives on that castration front, and I think it may be more in use in Canada than it is in the U.S. Certainly, it's more in use in Europe than it is in the U.S. The one that I think of, has the disadvantages of having to inject these older pigs twice. So you have the cost of handling those pigs twice, but you've also lost most of the feed efficiency advantage of the boars. I've heard about a system that's being developed, not yet approved, where you can do the injection when they're 3 days old. So from the point of view of the handling, I think that will be great, but you won't have that feed efficiency advantage.
Now, you mentioned at the start the importance of tryptophan metabolism. in order to get scatol absorbed into the bloodstream. Has any work been done to eliminate or reduce tryptophan metabolism or fermentation to prevent this scatol from developing in the gut?
So a number of people have worked with dietary manipulation to reduce the scatol presence. People have tried chicory root, chestnut bark, raw potato starch, etc. And it seems that they can, to me, my interpretation is that they can show that they've reduced it, but not necessarily to the level that it needs to be reduced for pork, for the resulting pork to be acceptable to the vast majority of consumers. And the same thing in terms of genetics. They work to identify genes that they might be able to select and have pigs that had less Boar taint as a result of genetics. And I think eventually one might be able to find pigs that actually have these deletions in this promoter region that we're targeting. But so far, it seems to me that the studies targeting the genetics have reduced Boar taint in some cases, but they haven't got it to the point where the vast majority of consumers will be pleased with the pork product.
So then, what do you then see as next steps for your own research? You mentioned consumer panels. Let's say those consumer panels give the thumbs up. How do we get to practical application?
After the consumer panel that's planned very soon, we're going to try to generate some animals that do have, we're actually in the process of generating them, do have the 7 base pair deletion on both chromosomes, as well as some animals that have the 16 base pair deletion in both animals. And they will have litter mates that are wild type. The litter mates do not have any deletions in the chromosomes. So we can make the very best comparison between the animals with the deletion and the animals without. And for those, we'll be looking at feed efficiency, growth rate, as well as the general health and consumer panel, androstenone levels in the fat, scatol levels in the fat, sort of a more thorough analysis. So after that, then I think one can start working towards how do we make this work in a practical setting?
Yeah, in other words, you want to have some more biological data, both on the production parameter side and on what's going on in the animals itself.
I've been trying to pay attention to what's important, but nevertheless, it's not a production setting.
Excellent. Well, I enjoyed this conversation. Is there anything you would like to add for our listeners?
So I guess I'd say that I've been doing the talking, but I have colleagues who have helped tremendously with this research. Dr. Crystal Yang, Dr. Yan-Hong Liu, In-Sung Park, the veterinarians who helped with the transfer that created the initial animals. And of course, funding is an essential part of any research.
Funding always helps. Yeah. Well, thanks for the conversation today, and have a good day.
Thank you.